Mice navigate a complex and dynamic social environment, exhibiting a diverse range of social behaviors like aggression, maternal care, mating, and the establishment of social hierarchies.
Look at my brain.
Intriguingly, recent studies suggest that mice also possess the capacity for emotion recognition, the ability to distinguish emotional states in other mice.
Hey, can you identify which mouse is displaying an emotion?
Yes, that one! While this fascinating ability has been established before, the underlying neural substrates remain poorly understood. In this work, we investigated neural mechanisms involved in emotion recognition in mice, focusing on the prelimbic cortex. To that end, we have employed a battery of 3 behavioral tasks, where subject mice are presented with a naive stimulus animal in one corner of the arena and another stimulus animal that has undergone a manipulation to induce either relief, stress, or social craving in the other corner. We found that both male and female C57 mice preferred emotionally aroused stimuli, except for the isolation tasks where only males preferred the socially isolated stimulus over the naive. To examine prelimbic cortex neural activity during social interactions with stimuli of varying emotional states, we used fiber photometry to record calcium signals during social preference, emotional state preference for stress, and emotional state preference for isolation tasks. Results have shown that long, but not short, bouts of interaction with emotionally aroused stimuli induce distinct neural activity patterns. Specifically, long bouts of interaction with the emotionally aroused stimuli induced a decrease in pyramidal neurons activity in the prelimbic, while long interactions with the naive stimulus induced an increase in activity. To gain more detailed insight into the activity of prelimbic cortex during social interactions with emotionally aroused conspecifics, we used chronically implanted neuropixel probes to record neural activity at the single-cell level. The results show a variety of neural activation schemes, ranging from strong excitation to strong inhibition in response to either the start or the end of investigation bouts in a stimulus-dependent manner. Each type of response characterized a different group of neurons. Typically, each group of neurons responded with excitation only during one of the 4 types of events. To understand how important the activity of pyramidal neurons in the prelimbic is for emotion recognition, we employed both excitatory and inhibitory optogenetic manipulations specifically to these neurons. We found that in emotional state preference for stress task, activating pyramidal neurons within the prelimbic at the first second of interaction bouts with the stress stimulus abolished the behavioral preference for the stressed stimulus, while activation of these neurons at the beginning of interactions with the naive stimulus preserved or even augmented the preference. Optogenetic inhibition, in contrast, yielded an opposite trend, where inhibition of pyramidal neurons within the prelimbic at the start of interaction with the stressed stimulus preserved the behavioral preference, while inhibition at the start of interaction with the naive stimulus abolished the preference.
C57 mice can recognize and differentiate between distinct emotional states of other mice. Interacting with emotionally aroused stimuli triggers unique activity patterns in PRL pyramidal neurons, reduced activity for aroused stimuli, and increased for naive ones. The PRL contains distinct neuronal populations that selectively respond at either the start or the end of an interaction, depending on the emotional state of the stimulus mouse. Pyramidal neuronal activity in the PRL is essential for typical emotion recognition-based behavior.